So, hello everyone, good evening. Am I audible?
Hi ma'am, good evening. Yes, you are audible.
Okay, I can see someone new. Tanu, have you joined for the first time?
No, I have joined this for the second time.
Okay. Okay, so let's start today's session. So, hello everyone, good evening. Myself, Rajal Bachawa. I'm your PMRF research scholar from IIT BHU Varanasi. I will be your teaching assistant for this particular course, that is host pathogen interaction, and the duration for which is January to April. And today is the week 7 lecture of this particular course. Till now, 6 week course is already available on the YouTube. I have discussed the assignment problem until week 5, and today we will be discussing the assignment problem for week 6. And all the videos are available on YouTube. If you have not watched, you can go there and check it out. And the lecture material for until week 8, I think, yes, it's for week 8, the lecture material and the assignment is released on the NPTEL. So, and our regular timing is every Saturday 3 to 5 p.m. until this week 8, up to the antigen and antibody course material is already available on the NPTEL, so you can check it out. So till in our last class we have studied about like toll-like receptor. We have solved the questions about toll-like receptor. In from the today's class we will be solving this RLR and NLR. That is the RIG1-like receptor and the NOD-like receptor. So have you studied these lectures from the lecture material available on the YouTube from Himanshu sir? Have you guys seen the video lectures?
Yes, ma'am.
Okay, so I'm hoping that you might be little bit familiar with these terms and these receptors, domains, and their pathways. So I would like to take a quick revision of all the week 6 material so that we can solve the problems of this week 6. So would you like to comment on what are these RLR sensors and RLR receptors? Like you have seen the lecture video, so you understood from there, or if you have any particular doubt, so we can start by taking your doubt, or otherwise I will discuss everything in a quick summary. If you have any doubt, then you can ask.
Ma'am, please summarize everything.
Okay. So okay, I'll start from very basic things and try to cover this complete course of week 6. Somebody's there. So these RLR sensors are the like RIG1 like receptors that you have already seen in the like previous lectures where we have discussed about RLR, NLR and TLR. There were three type of receptors of innate immunity. Like from here you can see that in week 5 we have studied about this innate immunity pattern recognition receptors and TLRs. We have seen about like what are these pattern recognition receptors and what are these toll-like receptors. And today in week 6 we will be seeing about this RLR, that is rig one like receptor, and the NLR, that is not like receptor. So these are the three type of receptors in the innate immunity. These are the pattern recognition receptors. Like these are present on the cell surface of the pathogen which are recognized by our immune system. So this RLR receptor is, these are the cytosolic sensors. I will clear it here. This RLR are the, like these are the sorry. These are the like cytosolic sensors, and they can detect the viral RNA and initiate an immune response. Like on viral RNA surface, they can recognize. and inform our immune system that yes, there is some pathogen invasion in our body. So these are cytosolic receptors, and they play a very crucial role in recognizing these RNA viruses, and they can activate this type 1 interferon system. Like these can activate this interferon 1. Type 1 interferon system they can activate. And like there are three major family of this RLR sensors, which one is the first one is this REG1, that is retinoic acid inducible gene 1. Like this is retinoic acid inducible gene 1 or DDX58. This is the first type of this RLR receptor. Second one is MDA5, that is melanoma differentiation associated protein 5 or IFIH1 receptor. And the third one is LGP2, that is its name is Laboratory of Genetic and Physiology 2. These type of receptors are coming in the family of this RLR, that is the RIG1 like receptor. So it have like these three type of receptors are different, like they are not same. These are the domains. These you can see these are the receptor domains. So every domain is specific for specific function. Like here in these we can see this orange color one. This is CAR-D, that is caspase activation and recruitment domain. I will write it here. This is card domain, that is caspase activation and recruitment domain. This is car domain, this orange one. This is your car domain, car D. And this one is your purple one. This is your helicase domain. Helicase, from some basic biology, you might have heard about this helicase that is responsible for unwinding of the DNA or RNA. So this is the helicase domain. And this blue one is CTD, that is C terminal domain. This is CTD. So I think it's not working. So it's like C terminal domain. And this fourth red one is like it is your transmembrane domain. I think it's not working. Just a minute. Just give me 1 minute. Can you guys hear me?
Yes, ma'am.
Okay, so I think it's hang or there is some network issue. It's stuck. So just wait for 2 minutes. I think it's stuck, so I will correct it and join back. So you please stay there. Is my screen visible now?
Yes, ma'am, it's visible.
Okay, I'm so sorry. It's stuck in between.
It's okay.
So what I was telling about, like there are like these are left. So I think you have heard the full forms or noted it somewhere. No problem. I quickly write it here. Like there, these are like RIG1 like receptor which have this first type family is RIG1. The that is the retinoic acid inducible gene 1. The second one is MDA5, that is melanoma differentiation associated protein 5. The third one is this LGP2, that is laboratory of genetic and physiology 2. And these are different in their domain content, which have different function. The first domain is CAR domain, that is the CAR stands for here caspase activation, caspase activation and recruitment domain. The second one is helicase domain, which is mainly responsible for unwinding and winding of this RNA and DNA. And this third one is CTD. That is your CTD is your C terminal domain. It is your C terminal domain. And this red one, which is absent in most of these family here, which is your transmembrane domain. Red one is your transmembrane domain. So this CTD or the C-terminal domain is mainly responsible for regulating the RNA recognition and activation. And this transmembrane domain, which is not present here in this RLR family, but it is also type of domain, so you should be aware of this. So like there are this four type of domain, and in here this LGP2, that is the Laboratory of Genetic and Physiology 2 family, we can see here that this car domain is absent here. So I think later on one of question is there, what is different in this LGP2 from all other domains? So this is the car absence of this car domain. So like there are these type of domain, and these are the three type of families of RLR sensor. In the coming slides, we have discussed in detail about these type. So this is the first introduction slide of this RIG1 type sensor. And the next one is your recognition of RNA viruses by this RLRs, like how the RNA viruses are recognized. So here we can see that these are this different type of viruses here, like encephalomyiocarditis virus and this Newcastle disease virus, this vesicular stomatitis virus, Sendai virus, and this there are like different type of virus, and this these RLR are present on the surface of these viruses. So this immune system can recognize these type of domain and activate the downstream signaling. Like when this viral RNA will enter the cytoplasm during any infection, then this when this will enter in any cytoplasm, then this RIG1 domain, it will detect this double-stranded RNA virus or any single-stranded RNA virus, whichever is present in specific virus. And then this, like this RIG1, I'll write it here. RIG1 can recognize both double-stranded RNA and single-stranded RNA. And this other family, that is MDA5, it can recognize this double-stranded RNA, which is long double-stranded RNA, and this can recognize RIG1 can recognize the short double-stranded RNA. So when this virus will invade in any host system, then these type of like this RIG1 and MDA5, these will detect the RNA viruses, and the activation of this RIG1 and MDA5 will recruit an adapter protein, that is this IPS1. And this IPS1 will initiate the downstream signaling in like two different pathways, that is this IRF3 oblique 7 or this NFK beta signaling pathway. So this particular signaling will be activated by this by the type 1 interferon production, and this NF kappa B pathway will lead to pro-inflammatory cytokine production, and the immune system will be activated, and they will function on this pathogen, and they will attack this pathogen. So this is the basic mechanism of this, how these viruses are entering. Like in short, what they have discussed through this pathway, that there are different family of viruses which will try to attack on this host, and on their attack, this RIG1 and MDA5 proteins, these family will detect these type of RNA viruses, like whichever is entering in our body, and they will activate this CAR domain and this IPS1 domain, which will ultimately initiate the downstream signaling of whether this IRF3 by 7 or this NF kappa B signaling. And on the activation of this IRF3 by 7, type 1 interferon production will start, and on the activation of NF kappa B, this pro-inflammatory cytokine production will start, and these will attack on the virus, and they will ultimately kill this virus. So this is the mechanism of how this RNA viruses are recognized by this RLR type of family. So I hope this mechanism is clear to all of you. Or is there any doubt? So you can ask.
It's clear.
Okay. For others also, if you have any problem or something is not clear to you from the previously available lecture videos from the NPTEL or this current session, you can ask. I will discuss that again in the detail. So feel free to ask. And this next one is the activation of this RIG1 receptor and its role in the viral RNA recognition, like how this is getting activated and this is recognizing the RNA virus. So see in this RIG1, we have seen from this here, the first slide, this RIG1 contain the scar domain, helicase domain, and the C-terminal domain. So this have a particular structure in this like some semicircular form, that is something folded. It is not a straight structure. So what happens here, like this, it have two car domain, that is car D1 and car D2, which are required for its downstream signaling. And it have here this one helicase domain, a big long helicase domain, which helps in unwinding of this RNA. And it have the C terminal domain, which is responsible for recognizing the viral RNA. So what happens here? This RIG1 will bind to the viral RNA through its C terminal domain and the helicase domain. So on this binding, the conformation will change, and this RNA binding will cause like some rearrangement in the structure, that its CAR domain will be exposed. And this exposure will lead to this interaction with other downstream domain, like this CAR-D will interact with one other domain, that is MAV5, and it will initiate the downstream signaling. And this MAB5, this activation, it will trigger the this again. Here we have seen this no IR of 3 by 7 and NF kappa B signaling. So this this MAVS, it will initiate this IR of 3 or IR of 7 and this NF kappa B signaling. And this signaling will start producing this pro-inflammatory cytokines and interferon, type 1 interferon production, and it will ultimately kill the pathogen. So like this is the mechanism of functioning of this particular domain of RIG1. Like virus will come and it will interact here like this with C-terminal domain and the helicase domain, and it will activate this CAR D1 and CAR D2. This will next initiate this MAVS domain, and it will activate these two type of signaling. First one is either IRF3 or IRF7 or NF kappa B signaling, and it will kill our pathogen. So this is the mechanism of RIG1 functioning. And next there is a like this Baltimore classification of by Baltimore classification we have different type of DNA, that is the type 1 double stranded DNA and some of the single stranded RNA DNA. There is so much combination, and from this classification this virus family is there, and here they have shown that which RLR will interact with which type of this DNA or RNA, single stranded, double stranded. So, this is a table, you can check it out from the lecture video. There is nothing much to explain in this. So, the next one is this activation of RIG1 by host RNA, like during any infection, how this is getting activated. So, here what happens that like in this HSV1 virus infection, that is the herpes simplex virus 1, this RIG1 is highly enriched with this. This is highly enriched with this particular RNA. So on this infection of this HSV1 in the body, this will RIG1 will have this like host cellular RNA. Wait a minute. Just a minute. Am I audible?
Yes, ma'am.
I don't know why the system is hanging so much today. So I'm so sorry for that. Yeah, so we were discussing the activation of this RIG1 by host RNA, like how it will get activated. Yeah, so in the infection of this HSV1, that is the herpes simplex 1 virus infection, this RIG is highly enriched in a specific host RNA. That is this RNA 5sp1141. So this is like this RIG is highly enriched with this host RNA, and this is a 5s rRNA pseudo gene transcript. So here, what happens when normally this RIG does not respond to self RNA because of the RNA modification, that is the methylation. So it will not respond to its own RNA. But during this infection, what happens? The cellular stress can lead to the unusual or mislocalized or misprocessed RNA, or it can lead to the like disturbance of cellular RNA homeostasis, And this can lead also to the like unmasked cellular non-coding RNA during mRNA, including like this miRNA. So what happened? This RIG1 domain will start responding to self RNA. So in this mechanism, what happens? Like this host stress lead to the RNA misprocessing or accumulation of non-coding RNA. And these RNAs will mimic the viral RNA structure. So what will happen when this RIG1 will bind to this host RNA? It will lead to the immune activation. And this can contribute to the autoimmunity if it is not properly regulated. So what will happen here in this case? Like this, these RNA will be misprocessed, or there can be an accumulation of non-coding RNA, and this RIG1 will bind to these host RNAs. In usual condition, it is not binding to its like own RNA, but during the infection, this RIG1 can bind to the host RNA, and it can lead to the autoimmunity. Because it will be not able to differentiate between which is the pathogenic RNA or which is its own RNA. Because this infection will cause the like misfolding or disturbance of RNA homeostasis, and this non-coding RNA will get accumulated. So this RIG1 will be not able to function properly, and it will recognize its own RNA, and it will lead to autoimmunity in the body. There is other mechanism of activation of this RIG1 by this host RNA again. Like in the last case, what was happening? The case of autoimmunity. And in this case, what is happening here? Like this RLR are getting mutated. Like mutations in these receptors can cause spontaneous signaling, and this can lead to immune response in the absence of viral infection, like even though there is no virus infection in the body, but then also this RLR, this RIG1 will get activated because this RLR are mutated. And in second case, what is happening here, there are like these two type of syndromes. One is this singleton Burton syndrome, and second one is this this Aicardi Goutieres syndrome. What is happening in these two syndromes? That this long hairpin RNA, which is derived from inverted repeats of this ALU elements, these have been suggested to activate the mutant form of MDA5. Like this MDA5 is responsible for recognizing the pathogenic this receptors, but in this particular syndrome the mutated form of this MDA5 will get activated, and it will recognize this the mutated form, and RIG receptor will get activated. So this is also a case of where RIG1 can be activated. And the next case is where this alteration in RNA metabolism can generate RLR stimulatory RNAs. Like the changes in RNA processing or degradation can produce RNA molecules that can activate this RLRs, and they can trigger the immune response. So, in the alterations in RNA metabolism, this can lead to the RLR activation. And the next case is of this mislocalization of mitochondrial RNA into the cytosol, which can lead to this MDA5 activation. Like in normal case, what happens? This mitochondrial RNA is present within the mitochondria, but if it escape into the cytosol, it can activate this MDA5, and which can lead to the immune response. So, like these are also some cases where this RIG1 receptor can get activated. So, like what we have seen from these two slides, that how this RIG1 can get activated from this host RNA, like by its own RNA. First one was the case of this misprocessing or this RNA homeostasis and these conditions. This can lead to recognition of RIG1 to its own RNA, and which leads to autoimmunity. The next case was of mutation in RLR which can occur in the case of even the absence of virus infection. So it can also lead to RIG1 activation. The next case was of these two syndrome, where this mutant form of MDA5 is activated. So it can also activate this RIG receptor. The third case was of this RNA metabolism. It can also activate this RIG receptor. And the last case is of mislocalization of mitochondrial DNA into the cytosol. So these are the classical cases where this RIG1 receptor can get activated. So now what will happen here if this RLR receptors are activated? After activation, what will happen? It will cause this downstream signaling activation initiation. Like what will happen? Like there are some proteins or some domains in this pathway. First one is this RIG1 and MDA5. These are the primary sensors of viral RNA in the cytosol. Like when the viral RNA will enter, first of all, these two will recognize the entry of viral RNA, and this RIG1 will recognize the short standard RNA, and this MDA5 will detect the long double stranded RNA that we have seen in the previous slides. And these will activate this MAV5, that is mitochondrial antiviral signaling protein. This MAVs, that is the mitochondrial antiviral signaling protein. These will activate MAVs, which is also called this VISC, that is virus induced signaling adapter, and CARDIF, that is card adapter inducing interferon beta, IPS1, that is interferon beta promoter stimulator 1. So, and it is located in the outer mitochondrial membrane. See, this is located here in this outer mitochondrial membrane, and it is also present on this peroxisomes. So once this RIG1 and MDA5, they will detect this viral RNA, they will interact with this MAVS, and they will initiate this antiviral signaling. Like they will signal to this peroxisome and the mitochondrial outer membrane. And from this MAVS activation, what will happen? It will recruit this TBK1 or IKK, that is the tank binding kinase, and these are like kinase proteins which are responsible for phosphorylation and activation of the downstream signaling. So this MAVS will activate these two kinases, which will phosphorylate these two key factors, that is this whether IRF3, IRF7 or NF kappa B. And these transcription factors will translocate to the nucleus, and they will promote the expression of this type 1 interferon alpha or type 1 interferon beta and some other antiviral genes. So from this mechanism, what is happening here, this pathogenic viral RNA is trying to enter in the cell, which is recognized by this RIG1 and MDA5, which then activates this MAVS, which signals to the domains present on this peroxisome, mitochondria, and mitochondrial outer membrane, and they will activate these kinase proteins, which is TBK1 and IKK. And these kinases will activate this IRF3 and IRF7 and an NF kappa B signaling. And this signaling will lead to their entry inside, and this will finally lead to the expression of this type 1 interferon and other antiviral genes, so which can lead to the killing of this virus. So this is the mechanism and the signaling pathway of this. RLR. And the next one is RLR pathway regulation, the proteins and the non-coding RNAs involved in this. Like this is a much bit detailed pathway and signaling, but the key points to keep in mind from this is that like this RIG1 and MDA5 will detect the viral RNA, but their activation is modulated by regulatory proteins and non-coding RNA. Here, what will happen is RIG1 and MDA5. These both are responsible for recognizing the viral RNA. So, there are some key points that you have to keep in mind. And there are several RNA binding proteins. Like you can see here, there's one are the RNA binding proteins which will recognize this RNA and they will activate some downstream the signaling pathways, and ultimately what will happen, like these will in the end they will lead to the production of this, here they will lead to the production of this interferon and other antiviral proteins, and which will ultimately lead to the killing of this pathogen. Like there are so many factors, so many proteins to keep in mind. Like here, this ubiquitination is also involved, which will mark this viral protein to further signal it to get degraded. And ultimately, in short, this thing will happen. What we have seen here, like these two families will get activated and they will ultimately lead to the production of this interferon. So, this is the detailed signaling pathway of that slide only. So next there is this RLR pathway regulation by this its post-translational modifications of this RIG1 and MDA5. So like this post-translational modification is very much important for the activation and inhibition of these type of proteins and receptors. So here what will happen, like there are some activating PTMs, there are some inhibitory PTMs in both RIG1 and MDA5. They have discussed in detail about here. Like sometime it will get like this sumoylation, like addition of sumo group, but sometimes it will get ubiquitinylation, sometimes it will get phosphorylation, and like there will be so many type of post translational modification. So ultimately what you have to keep in mind that there are some activating pdm which will promote their activation, like I will quickly write it here: ubiquitination, sumylation, phosphorylation, and deubiquitination. So these are the major type of PTM which will occur here, and depending on what subunit and at what stage this particular PTM is happening, their activation or inhibition will be decided by this. And there are some of the RLR mutations and disease associated with this. Like if particular RLR is getting mutated, what disease will be there? So you can check it out from this table. I hope till here this is clear. Like there are so much detail to this, and these factors and domains have so many functions, so it is not possible to remember each and everything, but summarize all the things on a paper so that you can know which domain is activating which domain, and by the activation of what thing, finally what is getting produced, like which type of interferon is getting produced, which type of other this antiviral proteins are getting produced, and how they are responsible for the pathogen killing. So in summary form, you should know about all the domains. Otherwise, like these pathways, you can't remember, and there is no need to remember these pathways. Just you need to keep in mind that there are some of the I'm sorry. Like there are some of the PTMs which are responsible for activation of this domain, and there are some which can inhibit this domains. And this also like which domain is getting activated, which domain is getting interacted with which downstream domain. So these things you have to keep in mind. So there is no need to remember all this in the detail. So I hope the summary is clear till here. Or you have any doubt or any confusion?
Yes, ma'am, it's understandable.
Okay.
Thanks.
Just a minute. So these RLR ligands are can be used in the therapeutics of this anti-tumor immune response, and like here you can see, for example, am I audible?
Yes, ma'am, you're audible.
Okay, there's a lot of network issue and the system is also hanging. So okay, there are this RLR ligands can be used in the therapeutics also. Like for example, this RLR agonist, which is identified, and this can activate the immune response against tumor. Like this is can generate an anti-tumor immune response, and it can also induce the cancer cell apoptosis. So these are the like some examples of which domains can be responsible for this. And there are some RLR domain which can lead to the immunogenic cell death through the damage associated molecular pattern release, that is this ICT DAM from the tumor cells. So these are the examples of this agonist. And there are also some this agonist which can induce the antitumor response. So these RLR ligands can also be used in the therapeutics. And also this nanoparticle deliver RIG1 natural ligand TPP double stranded DNA into the pancreatic cancer, and it can successfully seize the tumor progression. And also the co-treatment with this RLR agonist, it can lead to the enhancement of anti-cancer effects. And this can also induce immunogenic cell death and can lead to the inhibition of tumor growth and metastasis. So these RLR ligands are very much helpful in the this therapeutic development. And this RLR can are also involved in the immunopathogenesis, like the prolonged exposure to type 1 interferon environment can lead to an anti-inflammatory response, and inappropriate use of this RLR agonist may also result in an out of control inflammatory response. So this can also lead to the immunopathogenesis, and this high RIG1 expression is also associated with ovarian cancer. So these have both good effects and the bad effects of this RLR. So next one is this NLR. So till now we have studied about this RLR, and now we will start about this NLR, that is the node-like receptor. So from the very previous some sessions you have heard about this TLR, RLR, and NLR. There are three type of innate immunity response. So here this is NLR. These NLR are node-like receptor, and this node here is nucleotide binding polymerization domain. It is the node. So these are NLR. So what you know about these NLR, this node-like receptors?
First, they were discovered in plants, and they were called genes, as R stands for resistance for the
Mhm.
parasites and pathogens that were in the plants. And then it is conserved in all the animals. Like some homologs are also present in vertebrates and fishes and urchins. Then it is considered to be conserved, and yeah, there are two different functions. One is they sense, and they sense the pathogen and they activate the NF kappa B signaling. And the other is they are responsible for the production of interleukin 1 cytokines and also some apoptosis.
Yeah, very good. So you know quite well the introduction of this NLR. So what is the point of discussing? Like I'm less familiar with these topics, that's why I'm unable to explain you these in the detail. So till the previous lectures, the all the topic I have already studied in my bachelor's, masters, and I'm quite familiar with that. So I can frame the stories with that and make you understand. But these receptors and this NLR, TLR, these are there is some pathways, there is signaling, there is interaction, there is expression. So can't frame up stories with that here. So I can't explain these in detail very well. So I hope from the video lectures you are already have seen that and you are familiar with this topic. And so if you have any doubt, then we can discuss this. Like I can explain if you have any doubt at specific point, like how their interaction is working or basic things. I think you are aware of this. For the other students also who are connected, are you aware of these topics, some basics, or like you want me to explain this? Is anybody else also connected here? So, are you aware of this topic, the basic things, or you want to have a discussion on this? So, okay, no problem. We will go for discussion, short summary. Yeah, so this is the introduction of this NLR, that is the node-like receptor. These NLR were first identified in plants as a part of their antimicrobial resistance genes, which is the this R gene. And this NLR homologs exist in this vertebrates, including the primitive organisms like the sea urchins, and these are evolutionary conserved and they play a critical role in immune defense like and inflammation. And these like humans have this 22 NLR proteins while mice have 33 genes in the mice. And also this indicate the species specific immune adaptation. So these are the NLR that are the node like receptor. And this is the classification of this receptor. Like these are also present in human, mouse, and also in other organisms. And their domain organization is this, like we have seen in the somebody is there or something happened. Okay. So like this, every type of NLR family have a different domain organization. Like these are the different type of domains, and every domain have a different function. So this is the domain organization of all this NLR family receptors. This is the structure and function of this NLR. So your first receptor is this LRR, that is the leucine rich repeats. It detects the microbial component. The second one is this NOD, that is the nucleotide binding domain. It required for ATP dependent activation. The third one is this GARD, that is the gas-based activation and recruitment domain. It facilitates the protein-protein interaction. The next one is Next domain is this PYD, that is the pyrene domain. It is involved in inflammasome formation. And the next year domain is this FIIND, which is function to find domain. And the next one is BIR, that is baculovirus IAP repeat. So like there are different type of these receptors and domain. Like mainly there are this NOD1 and NOD2 ligands which are responsible for this NLR signaling. So what happens here? This NOD1, this detects the this IDEB in the gram-negative bacteria, and this NOD2 it detects the MDP in both gram-negative and the gram-positive. This only detect in gram-negative. It detects in gram-negative and positive. And these peptidoglycan fragments are released during the bacterial infection and are recognized by this NOD1 and NOD2 ligands inside the host cell. And the activation of these NOD1 and 2 it triggers the immune response, including the NF kappa B signaling and the cytokine production. And the mutation on in this NOD2 domain is linked with the inflammatory disease like this Crohn disease, so it can also happen. And these are the full names of this MDP, that is the muramyl dipeptide, and IEDAP, that is the intracellular or the gamma glutamide mesodap, that is the IDAP. These are the domains which are recognized by this NOD1 and NOD2 ligands. And this is the peptidoglycan structure layer of this gram-positive bacteria, in which this NAG and NAM, this cross chains are present, which is the N-acetylglucosamine, and NAG is the N-acetylmuraminic acid, and it is cross-linked by this L-alanine and D-glutamic acid, and also this MDAP is present there, and this D-alanine is also found there. So these are responsible for cross-linking this chain, this glycan chains in gram-positive and gram-negative. So this NOD1 will recognize here the IEDAP domain, and this NOD2 will recognize here the MDP domain of gram-negative and positive, and they will ultimately activate the NF-kappa B signaling and the cytokine production, which will kill these pathogens. So yeah, this is the similar mechanism of this NOD1 and NOD2. And this is the activation of inflammasome. So what are these inflammasomes?
They are a group of proteins which are involved in the activation of converting the inactive form of enzyme to an active form,
Yes,
especially procaspase to the caspase.
yes. So these inflammasomes are multi-protein complex that play a critical role in innate immunity by detecting pathogen and cellular stress. Their activation can lead to the production of pro-inflammatory cytokines. Like how these inflammasomes get activated when there is a trigger by the pathogen, or there is a danger signal, that is the like from the PAM or the DAMP that we have seen in the previous lecture. PAMP is the like this pathogen associated molecular patterns, and this DAMP is the danger associated molecular patterns. So this inflammasomes will get activated when there is a danger to the host cell. Like there are various type of this NLRs involved in the inflammasome formation. That is this NALP3, NR, NLRC4, NIP5, and these are NLRs involved in the activation of this inflammasomes. So there are different type of inflammasomes and their activation by like this NLRP1 will get activated by the danger signals of this lethal toxin MDP or ATP, and this NLRP3 will get activated by this type of vamp or temp. Like there are specific activation signals for different type of inflammasomes. And here this NAIP and NLRC4 will get activated by these danger signals, that is the flagenin or the road proteins. And this AIM2 will get activated by the cytoplasmic DNA. So there is different activation mechanism of different type of inflammasomes. There are various disease associated with this NLR mutation like this Muckle syndrome, familial cold, autoinflammatory syndrome, and this neonatal onset multisystem inflammatory disease. So this there is a loss of function mutation and gain of function mutation, and these two different type of mutations can lead to different type of disease. So what are these loss of function mutation and gain of function mutation?
Loss of function mutation is due to mutation, the specific function is being lost, whereas a new process or a thing is being added in gain of function mutation.
Hmm. So because of these type of mutations, there are different disease associated with this NLRs. So you can check out their names from here, like which receptor mutation will cause which disease. And this NOD2 is also associated with this Crohn disease. Because this NOD2 is mainly present in the gut immunities, and it is highly expressed in the this macrophages and dendritic cells in the intestinal lining, and it can detect the bacterial components and can trigger the immune response. And these macrophages and dendritic cells can use NOD2 to recognize and eliminate the harmful pathogens from the body. So, what will happen here? This mutation in NOD2 can impair the bacterial clearance and can increase the pro inflammatory cytokine production, which will dysregulate the immune signaling and it will contribute to the Crohn disease pathogenesis. There are some also other disease with this NLR mutation. The next topic is your DNA sensors. So, what are these DNA sensors and what is their role in the immunity? Like these DNA sensors are the proteins that detect the foreign or mislocalized DNA within the cell and they can activate the innate immune system. These are capable of detecting this mislocalized some DNAs in the body and they can activate the immune system and they can induce the type 1 interferon and pro-inflammatory cytokine production and they can also induce the programmed cell death as a response to the infection. Like because of the different type or different property of this DNA sensor, it can have a different effect, like because of what can happen because of its difference in its length or 3D structure or the sequence of cytotoxic DNA or the subcellular localization of DNA molecules or the methylation status of DNA or the association of histone and non-histone chromatin binding protein with the cytosolic DNA molecules. So these factors can affect the DNA sensor specificity. So, like this DNA is one will cleave the single stranded DNA and double stranded DNA in the extracellular space. And this cytosolic sensors can also activate this interferon signaling and inflammasomes, and while the nuclear sensors can regulate the DNA repair. This TLR9 can also sense the different microbial pathogens. These are the examples of different pathogens that can be detected by this TLR9. And this C gas can also sense the cytoplasmic DNA. Like there are different type of these DNA sensors which can detect the different pathogenic patterns or different receptors from the pathogens, and they can initiate the downstream signaling. Like some DNA sensors can also sense the RNA, or they can also detect the RNA virus infection. So let's quickly take the week 6 assignment questions. So I hope you all have submitted this already. So the first question of this assignment was, what makes LPG2 different from other RLR family members? As we have seen in the very first slide. So what is the
They
answer
don't
to
have any
this?
card domain.
Yes, the car domain is absent in this LPG2. Like we have seen here, the car domain is absent in these type of sensors. Now, next question is, what role does NF-kappa B play directly in the RLR signaling pathway once they are activated by RIG1 and MDA5? So, what is the role?
They are involved in the production of inflammatory cytokines.
Yes, how they are, why they are producing this inflammatory cytokines?
Transcription, through transcription.
Yes. So, the role of NF-kappa B in the RLR signaling is the transcription of inflammatory cytokine. And the third question was, which of the following is a distinct role in the IFI16 sensor compared to other DNA sensors like IM2 or C gas? What is the distinct role of this IFI16 sensor? Like in the just last slide, we have seen that it can sense both the DNA and the RNA. So this IFI16 sensor have a peculiar property of sensing both DNA and the RNA from the pathogens. So, what is the key role of loss of function mutation in NLRs? What will happen if NLRs have a loss of function mutation in them?
Causes immune deficiency syndrome.
So, why will happen this like this bar lymphocytic syndrome, like immune deficiency syndrome? Why will happen because of the loss of function? What change will occur there?
There is a severe reduction in CD4 T cells because of
Because of that, this immune deficiency
vascular.
syndrome.
Yeah.
Yes. So the next question is which of the following statement describe the implications of NLR mutations in human disease? Implications of NLR mutations in human disease.
They can be both gain of function as well as loss of function. And
Hmm.
NLRP3 are linked to a variety of inflammatory diseases like their mutation.
Hmm.
Then NOD2 mutations have been associated with Crohn's disease. Then genetic variation in NLR contributes to diverse immune responses and diseases susceptibility.
And why the other options are wrong?
Gain of function mutations are not linked to immune deficiency syndrome, but loss of function is. And
Yes.
as well as heightened immune responses are due to gain of function, and not all NLR mutations lead to inflammatory conditions.
Yes.
And yeah,
So the implications of this annular mutations in human disease is like these annular mutations can lead to both gain and the loss of the function mutation. And the mutations in this receptor are linked to a variety of inflammatory disease. Yes, we have seen that. And these NOD2 mutations have been associated with Crohn's disease, and the genetic variations in NLRs can contribute to the diverse immune response and the disease susceptibility. So, this is also correct option for this NLR mutation in human. The next question is which is not an adapter of RIG1 protein for downstream signaling?
TRAF6, which is an adapter molecule in toll-like receptor, but not
Yes,
in
not
RIG
in.
receptor.
Yes. So here we have seen from the starting slide that there are these type of domains in the RIG1 protein downstream signaling, but this TRAF6 is not present here. Rest four are present in RLR. The next question is which of the following statements are true about the activation process of RIG1 and MDA5 in the RLR pathway? So what they are asking about here about the activation process of RIG1 and MDA5 in the RLR pathway, like how it is getting activated.
Option A is not true because they're not always in active form,
Yes,
and their activation not only occurs in peroxisomes but also in the mitochondrial membrane as well as in the mitochondria.
yes,
And it does not only relies on the phosphorylation by MAVs but also by RIG1, like other
yes.
processes also
Other
occur.
also there.
Yeah. Um, and F option is also wrong because MDA5 is also involved in NF kappa B activation,
Yes,
and they are not anchored to the mitochondrial membrane.
yes. So the correct options are activation leads to the conformational changes exposing the scar and helicase domain that we have seen in the semicircular structure of this RIG1 activation, and they can recruit the adapter molecules with CAR domain for signaling. Yes, they can do this, and their interaction with viral RNA initiates the activation process. So this is also correct option. Next question is which one of the following statements best describe the function of ESC in the inflammasomes complex?
It recruits pro-caspase-1 through card-card interaction as ASC contains one card and one pyridine domain. And card-card interaction, there is a homotypic interaction between the caspase as well as the ASC.
Yeah, so this ASC will recruit the procaspase 1 through gut-gut interaction. The next question is, in the context of DNA sensing and type 1 interferon production, what role does the protein IRF3 primarily play? So why this option is correct and others are wrong in the context of DNA sensing and type 1 interferon production? The role of IRF3, they are asking. Like this IRF3 is responsible for it is a transcription factor for the interferon production. The next question is, which protein is characterized as an adapter that engage TBK1 upon DNA stimulation? So which protein is responsible here?
MITA.
So the correct option is here MITA protein. So these were the questions from the week 6 assignment, and till now these topics are already covered by Himanshu sir on the NPTEL portal, so you can check it out. So we have seen questions still here. This innate immunity receptors, that is TLR, NLR, and uh, NDR. So from the next session onwards, we will be discussing about this antigen and antibody and the innate and the adaptive immune system, which is the much interesting topic of this. And in today's lecture, there was so many type of this receptors, the families, the signaling. So it is little bit difficult to explain. So if it is not clear well to you, so you can let me know. We can have a discussion, or you can drop me an email. We can have that in the next class again. So I thank you everyone for joining till here, and I hope these things are clear, not explained very much clearly from my side for this week. So it was not a good session from my side, but we'll have a better session from the next week onwards, because the topics are different. We can frame up so much stories and can explain it to you in better way. So yeah, so any comment from your side, please let me
No, ma'am. Thank you for today's session.
know. Okay. Okay, thank you, Tanu. Okay, thank you students for joining till here. So you can leave. Thank you.
Thank you.
Okay.